So this is Dr. Szafranski. She is a fourth-year parasitology resident at the UTCVM through the National Center for Veterinary Parasitology. She's also a PhD candidate in UT's Comparative and Experimental Medicine program, where her main project focuses on Toxoplasma gondii in waterfowl. So a little different than Lyme disease that we're talking about today. So if you all have interest in T. gondii as well, I'm sure she's quite the expert getting ready to prepare her dissertation for that work. Her research interests include zoonotic parasites and the diagnosis and management of parasitic diseases in wildlife populations, as well as domestic animals. And today she's going to talk about the parasitology and microbiology crossover topic of Lyme disease, given that May is Lyme Disease Awareness Month. So maybe a little less on the human side she sort of talked to us about, but giving us a good overview here, and she'll be taking questions at the end. Again, feel free to put those in the chat. And with that, I'll hand it over to you, Nicole.
Oh boy, thank you so much. Thanks all for being here and for inviting me to talk. If there are any microbiologists in the crowd, I apologize. I study parasitology. And so if I say anything incorrect about the microbiome part, please correct me. But I'm excited to talk with you about ticks and about what I know of Lyme disease, mainly from a Southeastern perspective. I think Lyme disease has a really cool cross in One Health, where it truly encompasses all of the aspects we think of with physical, environmental health, with the health of our plants, animals, and people. And so I'm excited to dive into that a little bit today. And other disclaimer, if you hear my cats yowling in the background, I have closed them out of my office, and they usually get pretty offended when I do that. So if you hear a cat, my apologies. The dog will be perfectly behaved, but the cat's a little wild. Okay. So tick-borne diseases, I feel like we hear about these a lot in the media. We hear about them both on mainstream media and we hear about them a lot on social media. And I think that's a really interesting dynamic as we've moved into kind of this realm of social media, that we get a lot of good information, but we also get a lot of misinformation. And so as we go through the talk today, I'll ask you to think about some things that we might hear on social media. I don't know if any of you are users of Instagram, Facebook, TikTok, things like that. But I feel like every few months we get this, or at least I get, the algorithm's very good at picking up what I am interested in. These surges of Lyme disease, chronic Lyme manifestations, oh, there's my computer too. Tick-borne diseases. What do we need to do about Rocky Mountain spotted fever? Have you been infected by Lyme in the past and now you have chronic immune-mediated disease? So there's a lot of things that we see spring up, and different diets and exercise regimes and all of these things that people talk about in association with tick-borne diseases. And so we'll touch on that a little bit and see if those check out or not. And like I mentioned, it's a really cool One Health approach. For me, I'm in more of the animal health side, being a veterinarian and studying parasites. So I work mostly with the ticks that we're going to talk about today. But we do have a lot of interplay with how those ticks impact the animals themselves, how the pathogens in the ticks impact humans that they bite, and how the environment rolls into all of this. I think it's really fascinating that when we think about ticks, we don't just get to think about the tick itself. We get to think about what animals it's on, like the ticks that carry Lyme disease love to be on mice and they love to be on deer. And as those mice and deer ranges expand, the tick ranges expand because those animals take their ticks and other parasites with them. And then we see humans in those areas being infected where we didn't used to see infections. So it's a really cool interplay, not cool for those infected, but cool for people who study it, the interplay of how all of these things are working in this one little thing we call Lyme disease. So a quick history. I used to think it was hilarious when professors would put up the history of a disease or a process in class, because I was like, who cares? And now I'm the nerd on the other side of the table going, oh, shoot, I care. But I think it's really interesting how we piece together diseases, because it's not like one day where we just have this symptom and the next day we're like, oh, here's the disease. We got it. It takes decades for us to figure this out. I think our most recent example of this, which was actually really fast, was COVID. So with COVID, we had an identification of what it was, we had diagnostic tests, and then we had a vaccine all within a few months to years. That's really fast. And I think sometimes when us scientists are talking with the public, the public isn't really aware of how fast that process was and how that isn't really reflected in a lot of the other disease processes that we see. So a little bit about the history of Lyme, which I think is super cool. It was first described in Europe in the late 1800s, and it was simply described as a chronic skin disorder. They described this red rash, and it lasted for a long time. And that's pretty much all they said. We didn't have a way to treat it. It was just noted. In 1902, a term was starting to be used in the medical community in Europe called acrodermatitis chronica atropicans, which is a big old term. But essentially, it's in the skin, it's chronic, and we kind of have a weakening or an atrophying of the skin with redness and then skin type thinning. In 1909, there was a dermatologist in Sweden, and there was a young woman who was infected with a slowly expanding skin lesion. And this is where we get the term erythema migrans. And so erythema migrans kind of goes back to the Greek, and it literally means the spreading of redness. And so when we think of the bullseye mark for the Lyme disease, we think of that target where we get the tick bite. We have a whitish area, and then we have red spreading around. And that is where this term came from because the doctor noticed that was a red spot that traveled. So gets dropped off for a couple years. We don't hear much of it in the medical literature. And then Dr. Burkhardt in Switzerland in 1911 talks about a solitary cutaneous pseudolymphoma. So at this point in the disease recognition process, we realize that lymphocytes, a white blood cell, are the main player in this. And we usually see this one lump in the skin. So that's solitary cutaneous. So he thought, looks like a cancer, isn't a cancer, we have one lump. Finally, in 1913, we have that connection to a tick bite. There was this young 29-year-old woman who came into this dermatologist, and she had the spreading red rash. And he finally linked it back to she was bitten by a tick a few weeks before she presented to him. And again, it was dropped off for about 10 years. So we get these little pieces of the puzzle starting to come together, but people don't really know what it is. We just get that it's the skin lesion that happens. Finally, in France, we have our first introduction of the bacterial component. Couple of neurologists described a Borrelial meningo polyradiculoneuritis. And so this is inflammation of the meninges on the brain, so the covering of our brain, and then an expanding through our extremities type neuritis or nerve inflammation. And so now we have a link between a bacteria and the symptoms we're seeing, but we still haven't made that connection to the tick. In the 1930s, finally we get that connection. One of the doctors who was working on this goes, oh my gosh, we have a tick bite, then we have a skin lesion, and then we have these neurologic signs. So finally, we've put together the picture of what we think of today as Lyme disease, where we have a tick, bite, skin lesion, and then we get joint pain, and then we have nervous system involvement. And so as this goes on through the 1940s, we start getting the building of this description. We get more cases, we get a better definition of what these cases look like. And then finally, after World War II, we start treating these cases with antibiotics because we now have antibiotics that have been discovered to treat them with. And we realize that they are responsive to those antibiotics. So that's pretty much where we get from the late 1800s to 1940s. And then it kind of drops off the map. And it comes back finally now in the US, up in the Northeast in Lyme, Connecticut, where there's this little cluster in the 1970s of juvenile arthritis cases. And for the juvenile arthritis cases, originally they thought, this is just an immune-mediated response. But then they noticed that they were all clustered together and that they were happening at a more high rate than what you would expect for an immune-mediated process in that population. So shout out to the moms. There are these two moms in Lyme, Connecticut, who called the Connecticut State Health Department and were like, hey, something is happening here. A ton of the kids in our neighborhood are coming up with this joint pain. They're getting fevers, and we don't know what it is. So the Connecticut State Health Department put out a surveillance effort into Old Lyme, Lyme, and East Haddam, Connecticut, where they were looking for children that were affected by recurrent pain and swelling in their joints that had been preceded by a skin rash about 4 weeks prior. So once they had this group of children, they were like, okay, now we can kind of start narrowing down what's going on here. And they didn't know what was going on there. So they just termed this Lyme arthritis. And it was a little epidemic, Lyme arthritis, that's all we know. So as this is going on in Connecticut, Yale started working with patients that had these rashes as well. And some of these people with rashes would develop arthritis, and others would develop arthritis and neurologic symptoms, and then we would have some with cardiac symptoms as well. And so now we get the increase from Lyme arthritis into Lyme disease because we've gone from just one area being infected to multi-systems being affected. So now we have Lyme disease. We know it causes skin lesions, we know it causes neurologic problems, and we know it can cause cardiac symptoms. And then since it's antibiotic responsive, they're assuming there's a bacterial agent that is involved. Finally, so this is like 90 years after we started describing this, the vector is identified as Ixodes scapularis, which is this beautiful little hard tick down here at the bottom. I don't know if you all can see my mouse. Very beautiful. To me, a parasitologist, I'm sure most of you are going, that's disgusting. But yeah, Ixodes scapularis. So the mystery was solved. We have our etiologic agent, we have where it goes and what it does in the body, and we have who gave it to whom. So Dr. Bergdorfer, which may sound familiar from Borrelia burgdorferi, is who we have to thank for this. So Dr. Bergdorfer was a fantastic researcher. He was actually looking for the causal agent of Rocky Mountain spotted fever in ticks. So Rickettsia rickettsii, which is down here in the bottom left, we have a little white blood cell, and all of the little dippy dots that we see around it that are purple, those are the rickettsial agents, a different type of bacteria-like organism. And he was looking for that. And so Dr. Bergdorfer was investigating ticks. And by doing that, he was taking, if you can imagine little ticks, he was dissecting them, and he was pulling out their salivary glands, their gastrointestinal tract, so their midgut and their hindgut, and then their reproductive tract. And he was staining all of those and looking for a bacterial agent, which if there are any grad students listening, you know how painful it is to do that type of very fine work, just hours and hours and hours. And this is what he was doing. He was dead set that he's going to find Rocky Mountain spotted fever. He's going to connect the dots and build that puzzle. He didn't. So he started looking for it in a different tick. Instead of Dermacentor variabilis, this tick up at the top, it's beautifully ornamented, has little headpiece. We commonly think of it as the American dog tick, super common on our dogs and cats. He started looking at other ticks in the area, one of those being Ixodes scapularis, or the black-legged tick, or sometimes we refer to it as the deer tick. And lo and behold, as he was dissecting these ticks out, he finally found something. It wasn't what he was looking for, but he found something. And he found these long coiled spirochetes in the midgut of the tick. And he was like, well, that's interesting. And he started looking back in the literature, as every scientist does with our lit searches. And as he was doing that, he found the links between these cases in Europe. And he also saw that spirochetes were being isolated from blood, skin, and CSF cerebrospinal fluid of patients affected by Lyme disease in the Northeast. So all of the pieces finally came together, and we had the causative agent that was finally named Borrelia burgdorferi in memory and thanks to him for all of his hard work. And this is what it looks like here down at the bottom. It is truly a little spiraled bacteria, very cute. Causes a lot of problems for being so cute. But our mystery is solved, and now we have a disease, and we know what's causing it and how this is all coming together. So the pathogen itself, these are immunofluorescent stained bacteria, but Borrelia burgdorferi is a gram-negative spirochete bacteria. Its main route of transmission is tick bite. We think of it either through the black-legged tick, Ixodes scapularis, or the western black-legged tick, Ixodes pacificus. And we do have some rare case reports of transplacental and transmammary transmission. So transplacental being from mom to offspring via the blood supply that's going back and forth between her and baby. And then transmammary is actually through the milk supply from mom to baby. So it doesn't happen often, but it can happen. And thankfully, one blood-borne pathogen that we don't have to worry about being transmitted via blood transfusions is Borrelia. So right now, we don't have any evidence that this gets transmitted via a blood transfusion, which is great news for the medical community because we didn't start checking blood for pathogens until pretty recently in the time of medical history. All right, some unique features of Borrelia. It's really cool. It has this outer surface protein. It has quite a few different types of them, and they're the markers that we can actually look at to follow the life cycle of the Borrelia from the tick invertebrate host into our mammalian host. So the outer surface proteins, what happens, our little unfed tick is starving. It gets on a mammal, it starts feeding. And as it's feeding, we get a change in that outer surface protein expression. So initially, the little tick has OSP-A, which is going to help the bacteria itself stick into the tick gut, and it's going to allow the bacteria to multiply in that tick's gut. Tick will go along, it'll bite a mammal of some sort, and it'll start feeding. As it's feeding, the blood is going to finally hit its digestion tract, and the OSP-A is going to kind of calm down, and the OSP-C, or the outer surface protein C, is going to kick up in regulation. And that's actually going to stimulate bacteria to leave their safe little place in the tick's stomach and head up towards its salivary glands into the great unknown. But this is how it gets to its next host. So OSP-C, Borrelia goes to the salivary glands. Eventually, it'll get into the host. So as the tick is feeding on the host, it's releasing a lot of salivary secretions. And in those secretions is the Borrelia organism. And then it gets into our mammalian host and the disease continues. So for pathogen to host and how this works, Ixodes ticks are unique in that they're a 3-host life cycle. They have a 3-host life cycle. So what that means is between each life stage of the tick, the larva, the nymph, and the adult, it'll feed on a mammal, it'll fall off of that mammal, it'll molt into its next stage, and then it'll get on another animal. And this helps the transmission of Lyme disease greatly, because if this little larva here picks up Lyme disease, or picks up the Borrelia organisms, it can carry it and drop it off to animals in the nymph, as it's feeding in the nymph stage and in the adult stage, to a variety of different animals. One thing to note is that we have, in parasitology, we have transstadial and transovarial transmission in ticks. Transovarial would be from mom tick to the eggs, and then the eggs hatch and those larvae come out into the world already infected with whatever organism they're going to be passing on. And then we have transstadial transmission, which is once they're out in the world, the larva can pass on an infection they've acquired to the nymph, and the nymph can pass it on to the adult. So with Lyme disease, thankfully, it does not transmit transovarially, so we don't have to worry about mom passing it to babies, but we do have to worry about a tick that gets infected, passing it on through each of its life stages. So it could get infected as a larva, infect a new animal as a nymph, and infect another animal as an adult, which means we have a longer period of time for that tick to infect more than one animal. So as the bacteria are ramping up inside of the tick, they're decreasing that outer surface protein A, they're increasing outer surface protein C, they're making the long travel up into the salivary glands, and finally they enter their new host. And lucky for us, this whole process takes about 48 hours. So you'll hear medical professionals, you'll hear infectious disease specialists talk about the importance of removing ticks quickly once they're attached to you. And part of the reason is the sooner you can remove a tick that's attached, the less chance you have of acquiring whatever infection they may be carrying with whatever pathogens they have inside of them. So if I get bit by a tick and I remove it after 6 hours, I'm taking away that chance of me getting infected with Lyme disease if it's an Ixodes tick, because it doesn't have that full time to decrease those proteins, increase those proteins, travel up to the salivary glands and get into that next animal. However, it is noted that there are certain diseases that pathogens that transmit very quickly. So things like Rocky Mountain spotted fever, in 30 minutes after a tick bite, it can be transmitted from tick to its host. And so it's really important that we check ourselves for ticks and we remove those ticks as quickly as we can, both from ourselves and from our pets and other animals. All right. So I had to talk about the ticks because I can't help myself, even though it's Lyme disease. The ticks are important because without them, we wouldn't have Lyme disease. Or we probably wouldn't find another way to get in there. The black-legged tick is the main vector of Borrelia, and we can see its distribution here in the US. It's primarily east of the Mississippi, and we have some really big concentrations up in the northeast of the country, in the Midwest, particularly the Wisconsin area, and then down in Florida. And so, as I'll show you in the next couple of slides, the prevalence of Lyme disease follows this pretty well, because that's where our vector is, and that's where our hosts live. Similarly, we do have sporadic cases that happen along the West Coast, particularly in the Pacific Northwest, kind of down that California coast. And these, we don't see Ixodes scapularis, but we see a cousin, Ixodes pacificus, out on the West Coast. And they can also transmit Borrelia pathogen. So we do see here our human cases of 2020, where our very dark greens are the higher groupings of cases. Wisconsin's a hotspot, and again, the Northeast is a hotspot, but we do have some along the West Coast, and those would be transmitted by those Ixodes pacificus. One thing to note that I always find kind of funny, not in a funny haha way, but in a funny ironic way, is that we have cases that we see up here, and then we have some in Florida. And now we normally don't see Ixodes scapularis naturally in high numbers in Florida, but we do see a lot of snowbirds that take their pets and themselves down to Florida every year. And so part of this Ixodes pathway could be people taking their pets and their ticks down into Florida, spending a lot of the time there in the winter. And now we have a very good Ixodes population down south that is also able to transmit the disease. The wonders of modern medicine have given us fantastic tests to see if we have Lyme disease or evidence of being exposed to Lyme disease. And thankfully in vet med, we have some fantastic resources that compile all of this data for us now. So there is a Companion Animal Parasite Council. It is a fantastic resource that I highly suggest if you have pets or if you're a vet, you look into this website. It has tons of information about different parasitic diseases and the testing and treatment and maps of the prevalence of these things across the US. But one thing that it shows that I want to highlight is that it follows the human markers, the human cases of Lyme disease very well. So we have a high prevalence up here in the Northeast, again, the Wisconsin Midwest area, or Florida. We do see some in Alaska from those Ixodes pacificus. And for us here in the Southeast, maybe 10, 20 years ago, we probably would have been in North Carolina, South Carolina, Tennessee, this really light orange where cases weren't really prevalent. But we're getting a higher incidence of cases in the Southeast over the years because the wildlife are moving, the ticks are moving further south, our animals are traveling more. And as all of those things happen, we see this disease traveling as well. And so this data comes from IDEXX. And so IDEXX is a very common diagnostic company that we use in vet med. And we can see that of the 11 million dogs that were tested for Lyme disease, over 400,000 of them were positive in just 1 year. One thing to note with Lyme disease seroprevalence is that we are looking for antibodies in the blood. And so antibodies in the blood don't necessarily equate to an active infection. It just means that that animal has seen that pathogen and mounted an immune response to it in the past. So it doesn't mean that we have over 400,000 dogs running around with acute Lyme disease. It just means that this many dogs that were of those that were tested have antibodies against Borrelia bacteria. And a very cool thing that one of my lab mates is working on. So Dr. Eliza Baker graduated from UT's vet school, very recently defended successfully her PhD from the CEM, and she did some fantastic work with zoonotic diseases in mesocarnivores. And of those, she looked at coyotes in particular from Tennessee and South Carolina. She tested for a whole variety of parasitic diseases, but within those, she did test for Lyme disease. Again, using that IDEXX SNAP test, which is a really convenient test to use. And of the 65 coyotes that she tested from Tennessee, 28 of them were found to have antibodies against Lyme. And then from South Carolina, 52, so we got pretty close to the same numbers, only one of them. So we do see that consistency and trend of it coming from the north to the south, and it hasn't really gotten into our southern South Carolina, Georgia, those type states yet. But I think over the next few years, we'll probably see it increasing. Another thing, part of her project was looking at the actual ticks, because looking at the vertebrate host is always a good thing, but we want to take that back and follow the full epidemiological picture. So she looked at Ixodes scapularis. I don't believe that she dissected out each little individual organism. Nowadays, we know where it lives, so we don't have to do that. We can just crush up the tick, extract the DNA, and test for the pathogens that we want to know. And using real-time PCR, she found Borrelia DNA in 4 out of the 86 ticks that she tested. She got really good sequences back from 3 of those, and she found 2 genotypes for that OSP-C, that protein that's getting the bacteria up to the tick salivary gland, type A and type N. I don't know much about the genotypes, the different genotypes within Lyme, but if anyone else does and they want to share that at the end of the talk, I am super happy to listen to it because I didn't know that they existed before a couple days ago. All right, swinging back to humans, prevalence and incidence. In 2019, we had over 34,000 confirmed and probable cases. And one thing that's really interesting is that the incidence depends greatly on geographic location. So if we look at the incidence, which is the number of new cases over a period of time, we don't really see any in the Great Plains. We don't see much in the West. We see a lot in the Northeast, which makes sense. That's where those ticks are, and that's where we find Borrelia organisms. Similarly, in the dogs, the seroprevalence matched up with humans very well, where we had extremely high prevalences in endemic areas, upwards of 90% in the Northeast. And then in Southwestern states, we had very little, so lower than 4%. Age groups are another thing to mention. We see an uptick in the youth, and then we see another uptick kind of in the middle adult, nearing towards retirement age. This could be because those are the age groups that are spending the most time outside. As kids, we love to go outside and play. And then once we get into our 30s and 40s, we're getting tired of the work life and we want to make sure we're spending more time outside, hiking, golfing, what have you. And we get a little dip here where we're in our working years. And then likewise, Lyme cases per year, they were only reporting from the CDC confirmed cases. They have now added probable cases to that. So what that means is confirmed cases have to match all of the case definitions. So for Lyme disease, we would have to have rash, we'd have to have serology, we'd have to have a positive PCR. And sometimes cases are really highly suggestive of a certain disease, but don't meet all of those requirements to fit a case definition. But now they're starting to include those as well, just so we can have an idea on if certain testing wasn't available or other things like that, that these are still probably true cases. We just don't have all of the information to call them a confirmed case. All right, signalment. So if we look at the writing, it looks like young males and adult males are most likely to get Lyme disease. It's true to a point, but if we follow the graph, the chart over here, it's not that different. Statistically significant, but not practically significant. Like anyone can get Lyme disease if you're outside and you get bit by an Ixodes type tick. And then for dogs, all breeds are susceptible. We usually see it in younger adult dogs, and we usually see it in lifestyle dogs that are more outside. So if we have hunting dogs, if we have dock diving dogs, if we have dogs that live outside for most of their life in a fenced yard or on a farm, those are the animals that we're seeing it in simply because their risk to exposure is higher. In humans, what do we actually see with the disease? We see an acute disease that can last up to a few months, and then there's discussion of a chronic disease with Lyme. For the acute disease, about 70% of the people that are infected with Lyme disease have that erythema migrans, that rash. And so we see where the tick bite would have been, we get an immune response, the inflammatory cells are coming in, it looks reddened, it's warm. And then as the bacteria is spreading out to go to different places in the body, we get that inflammatory reaction kind of following the bacteria. And so that leads to our traditional target lesion. In about 30% of cases, we will see arthritis, and then lower percentages, but we do also see neurologic deficits such as Bell's palsy, radiculoneuropathies, meningitis, encephalitis, and cardiac issues like arrhythmias. An important thing to mention is that historically, a lot of our diagnostic guides, especially on the human side of things, were based off of those of European descent. And so when we think of the bullseye rash, if you are of European descent, you usually typically think of this red target lesion. It's not always that. On different skin tones, it can appear very differently. And not only that, that target lesion is like a textbook definition of what Lyme disease presents as. And as we all know, as scientists, what we see in the textbook and what we see in real life are not the same thing. And so we want to be aware that if we have history of someone with a tick bite and then they have any kind of rash surrounding that bite, it's worth investigating. It's not always going to pop up as that beautiful target lesion. Okay. And then chronic Lyme. Chronic Lyme, I know there's a lot of controversy about. In the animal field, we do know that symptoms or clinical signs can last for many months. They are usually responsive to treatment, but they can last quite a while. The one thing that we don't know for sure in the human side of things is how closely related a history of a Lyme infection is to an immune-mediated disease in the future. So we do know that immune-mediated processes can happen within a Lyme infection, but how much of that is chronic, being years down the road where we're still seeing joint pain, if that's related to Lyme disease, is a little bit unclear. And if anyone on the human side is there and can correct me or discuss this at the end, I would love to hear it. But at least from the vet side and what I know is we don't have a really strong argument yet for years down the road chronic Lyme disease. But then again, it took us 90 years to figure out what Lyme disease was. So who knows, in 10 years, we may have a great description of it. All right, the things we first see in a human after being bit. We start to see clinical signs on average about 7 days after the tick bite. We can see it as early as 3. We can see it up to a month later. Fever, chills, headache, fatigue, lethargy, just feeling ugh, because your immune system is responding to that foreign material the tick is putting in you and its salivary stuff. We also see muscle and joint pain and lymphadenopathy, so swollen lymph nodes. And of course, that rash is very common, the erythema migrans, that uh, red rash. Later symptoms, so this could be days to months. We could see headache, neck stiffness, additional rashes. In some of the historic cases, they have rashes that creeped along the person's body all the way up to their ankles, to their neck. So it can be a very large rash. Neuromuscular symptoms, so the Borrelia can get into the nervous system, and as part of the immune response to fight off that pathogen, we can see signs associated with it. So we can see facial palsy, or that is the cranial nerves being affected. So sometimes Bell's palsy is where like one side of someone's face will start to droop because the nerves have lost that control to keep those muscles contracted. We can also see neuralgia or nerve pain, numbness and tingling, basically a lot of neuromuscular symptoms that can also be seen in our joints, in our bones, in our muscles, in our tendons, where these patients are just incredibly painful. A lot of the time they can't pinpoint the pain, but they do know that like one day my left knee is swollen, the next day my right knee is swollen, and then on Thursday my elbow hurts. So it's kind of this moving joint pain. Less common, but we can see cardiac arrhythmias and meningitis and encephalitis, so inflammation of the meninges and the brain itself, trying to fight off this pathogen. And a lot of these symptoms are actually caused by antibodies of our body trying to do the right thing. So they're trying to fight off the pathogen. They're attaching to the antigen of the bacteria, so pieces of the bacteria, and they're creating these antigen antibody complexes that travel around the body and sometimes get stuck in places they're not really supposed to get stuck and slow things down and cause even more inflammation. So in animals, more of my milieu, dogs, of the 100% of dogs that are infected, only 5 to 10% are going to show clinical signs, which is pretty wild. So if we think of that 400,000 plus number that we saw on the other slide, only 5 to 10% of them are going to be ones that show clinical signs. And who knows of that many, how many are actually going to show up to the vet clinic with their the client saying, hey, my dog's limping, or hey, my dog doesn't feel good. So things we see in dogs, lymphadenopathy, again, those swollen lymph nodes, this can be all over their body. It usually can be closer, at least in the beginning, to where the tick bite was. We can see joint swelling and shifting leg lameness. And this is one that's very funny if you're a vet student and you go in and you go, oh yeah, it's definitely limping on its right front leg. And then, the vet rechecks it in a few days and they come in and go, no, it's its left front leg. And you're like sitting there going, oh, come on, I know what I'm talking about. It was limping over there. But it can literally shift what leg is lame because the immune system is going to flare up in different spots. We can also see hyporexia, or a lack of willingness to eat, and lethargy, which can lead to muscle loss and wasting. And then the big one that we think of with dogs with severe disease is glomerulonephritis. So this is inflammation within the kidney, within the nephrons, in these little areas called glomeruli that help filter our blood. And this is where those antigen antibody complexes can get stuck. And so they're a little bit bigger than the blood that's normally passing through, and they don't pass through as well. They kind of clog up the system, and we can get a really severe immune response there. We also see Lyme disease in horses. We typically think of a rapidly progressive uveitis or inflammation in the eye leading to blindness. And if that continues and it continues to affect more of the central nervous system, we can see neuroborreliosis, which is where the Borrelia is within the nervous system. We can see muscle atrophy. We can see cranial nerve deficits like we would see in people. We can see ataxia or weakness and paralysis, and we can even see behavioral changes. So with horses, this can be a bit scary because you have a 1,200-pound animal that is wobbly and having shifting leg lameness, and they can also get something called hyperesthesia. And this is essentially an overreaction to touch, or our sensory nerves are super excited when we touch. So if I just walk over and normally put my hand on a horse, they might twitch, but they're not going to do much. One of these horses that has acute Lyme disease, if they're experiencing the neurological side effects of this, if I put my hand on that horse, they might buck up. Like it's such an intense, overwhelming feeling of pain because the nervous system, due to the immune response, is just overactive. And so with horses, this can be pretty severe. Thankfully, we don't see it super often, but when we do, it definitely needs to be treated quickly. And then for other animals, we see Lyme disease. We don't have very good descriptions of it. Thankfully, we don't often see this in cats, but we can see positive Lyme tests, at least antibodies, in a lot of different animals. So treatment and prevention things to do. Thankfully, after the dawn of antibiotics, we do have some effective ones for Lyme disease. And so our go-to is doxycycline. It is part of the tetracycline family. It works very well. But one thing to not forget is supportive care. So be it a human or an animal, they hurt. And when they hurt, we want to address that pain. So for supportive care, if an animal or a person is experiencing the muscle loss, the weight loss, the hyperexia, not wanting to eat, good nutritional supportive care is really important. Similarly, if they're dehydrated and lethargic in that first little bit of fever, we want to treat the fever within reason, give them some supportive fluids and things like that. And then the most important is the joint pain. When people say they're experiencing this nervous system pain and this joint pain and muscle pain, that's a really legitimate symptom or clinical sign in our animals that we need to take seriously and make sure that we are working on the comfort of our patients while we're addressing the Lyme disease picture at large. So steroids are probably not a great idea since we're treating something bacterial, but anti-inflammatory non-steroidals are a really great option to keep animals comfortable. Sometimes people will use gabapentin as more of a nerve pain relief, but as they all say, I'm going to say it wrong. An ounce of prevention is worth a pound of cure. There it is. So the biggest thing we want to do for Lyme disease is prevent. That is literally the best thing that we can do. So in terms of prevention, knowing where you are geographically is a huge thing. If you're in the middle of Idaho, you're probably not going to see it. You can worry about other tick-borne diseases, but not necessarily Lyme. But knowing where you are, knowing if these ticks are in that area, and then things you as a human can do, wearing long pants and having the awesome look of tucking those long pants into your socks. So when you were hiking, you were the cool one that has the tube socks on and little black dip dots along your socks because all the ticks are getting stuck there. Wearing light colored clothing. So one thing that ticks absolutely love and use to find their hosts are heat, carbon dioxide, and vibrations. So if you're trundling through a trail and you're wearing black clothes and you're sweating a bunch and you're really hot and you're huffing and puffing, you're giving that tick everything it needs to be able to find you. And so what we want to do when we go on hikes, when we go play outside, is we want to make sure we're doing things that reduce that chance or that risk of that tick finding us and attaching to us. So light colored clothing keeps us cooler. It also looks more like the sky than the ground, so they're less likely to climb up us. Keeping your lawns mowed, or at least avoiding the areas of tall grasses and leaf piles. Staying on the marked trail. A lot of people want to go wander off and look at the cool mushroom. Please don't. Use your iPhone, take a picture, zoom in. You don't need to wander off the trail to see the cool things. And then wearing tick repellent or some type of arthropod repellent. So if you're an adult, DEET, then 20 to 35% is great. If you're a child, you want to use less of a heavy concentration of DEET. And then we also have the pyrethrins. So for animals, tick checks, huge thing. Also, just year-round prevention. We have a lot of options compared to what we used to have years ago. We have oral ones, we have topicals, we have collars. They last anywhere from a month to up to 8 months. So it's really working with your veterinarian and finding what will work for you and your pet. But these can be great, easy ways to avoid a lot of negative consequences down the road. And then there is a Lyme vaccine. Um, its efficacy is okay. It's mostly based off of lifestyle. So if you have a small dog that you never take outside and you always carry it in your little handbag, it probably doesn't need a Lyme vaccine. But if you have a hunting dog and you're running around with it all the time and it's running through the woods and training raccoons, it's much more likely to come in contact with ticks. So that's one that's just a risk-based. Talk to your veterinarian and figure out what works for you. We love that we have a vaccine that works, but we also don't want to unnecessarily vaccinate animals that don't need it. And then horses, we don't have anything great. We do have some arthropod repellent sprays that are made of like pyrethrins and things like that. You usually have to spray them every day on your horse. You can treat the fields that they're in, but that has a lot of environmental consequences. So frequent brushing and then just looking for ticks are the two biggest things. This picture up here above the little gentleman showing us how to dress will show you a horse with some ticks attached. And if you're not looking closely, you can miss the ticks really easily. Horses have fantastic coats, but they also hide ticks really well. And then again, dogs, we don't want our dog looking concerned like this. We do want to make sure we are checking them for ticks as well and removing them. Big important things when you're removing ticks is to remove the entire tick. So when we think about a tick, we have a little headpiece that kind of dives into us and attaches, and that's where it's going to take its blood meal, and then we have the rest of the tick. And if you just pluck the tick's body off, I mean, you have killed it, so that is in part successful. But you left the headpiece attached, and when the headpiece is attached, the bacteria and all that are in the salivary glands can still pass to us. So we want to make sure that we are removing the tick in full all at once. We want to make sure that when we are removing that tick, we are not squeezing its head too much because we can actually squeeze and shoot the salivary stuff back into the animals. We don't want to do that. There are some awesome videos on YouTube if you're okay watching something a little gross about how to properly remove ticks. You can use tweezers, hemostats. I usually use my fingers. There are these tick removal devices that kind of look like a little Y that you slide into the tick or underneath the tick between you and the tick and pull. Those I have not found to work very well, but if they work great for you, that's awesome. And I guess the last little bit of advice is from a podcast that had a tick biologist on many years ago. And that tick biologist's advice was to check your crevices. So when you go outside, once you come in with your pets, please check everybody's crevices. Ask first if it's another human, but we want to make sure that we just don't have the ticks on us. That's the easiest way for us to prevent Lyme disease, is just making sure the ticks don't have a chance to get on us, or if they're on us, they're off of us very quickly. So I think with that, here are some of the references I used, and I am happy to answer any questions that you all may have for me.
Fantastic. Thank you for that awesome presentation.
Yeah,
We'll open it up for questions. I don't know if anyone has anything. I don't see anything in the chat yet, but feel free to unmute and ask your questions or post up in the chat, and I'm happy to read them.
yeah, I'm happy to hear from any human side of people who are probably chuckling at me for my lack of human knowledge.
That disease that horses get, that's
Mhm,
where they get spastic.
mhm.
Um, that's a different parasite, isn't it? And
So there are quite a few parasites that can affect the nervous system of horses. One of the
I'm
ones we
just
most
wondering,
say again?
I was wondering if that's Lyme does that too, that where they get spastic paralysis, or if it's just
Yeah.
other parasites.
Not so much the spastic paralysis, but that hyperesthesia where they get super touchy to like they're just overly neurologically stimulated when they feel that touch sensation. But normally we don't see a true spastic paralysis where the horse is in a stiff, can't move type sense of being.
Nicole, I've heard, maybe you can debunk this or
Oh boy.
confirm. That the role of lizards in transmission in the South, like ticks are getting on lizards and lizards don't transmit Lyme very well. So like lizards are protecting us here in the South. Is that a
Oh,
real
I
thing?
love it. All right. So, A, lizards are underrepresented in the amount of research we're doing with parasites. But B, yes. So when we think about how the disease is transmitted as it goes from the tick to the little mammal to the bigger mammal to us, we kind of think of lizards as a stopping point, because the tick will get attached to them. It can technically, like the Borrelia may pass through its salivary glands, but it's not going to be very effective. And so we think of them as kind of dead end hosts where that Lyme, that Borrelia is going to stop. So as we see Lyme more commonly in the South, given our prevalence of just reptilian species, they're kind of acting as a buffer for us as humans and other mammals. And so it's just yet another reason to protect our reptilian species.
Awesome. That's good to know. Yes, another
Yeah,
reason we should love them more.
I agree. Okay, I see. Oh, alpha gal. Ooh, that's a good one. So alpha gal is, I see a comment in the chat, is a, oh, it's really interesting. So in tick saliva, there are these enzymes, and they kind of closely look like, at least to our body, the breakdown of red meat proteins. And so if a person gets bit by a certain tick and their body responds enthusiastically, let's say, to that tick bite, that person down the road can actually develop a hypersensitivity to the red meat proteins that look very similar to our immune system, to the salivary compounds of the tick bite. And we don't have a good A, we don't have really good diagnostics for this, but B, we also don't have really good treatment for this. So those people are pretty much stuck not consuming red meat, at least we think for the rest of their life, at least currently. But yeah, that's a serious one. Okay. When our pets are treated with tick preventive chewables and drops, I observe that ticks still bite the animal and then die. I often find dead ticks attached, but I have no idea how long they were there. Is there a risk that these ticks were also able to transmit the disease? Oh, fantastic question. So normally within the FDA approval process, whatever, and the EPA, if we're using a topical, whatever product we are using, they have to kill so many ticks within so much time. And so most of the companies that are making these FDA or EPA approved products, they will start killing the ticks within 30 minutes, and usually most of them are killed within 24 hours. And so most of the time, the chance for Lyme disease to be transmitted is really, really low. Now, that said, we do have biological variation where every once in a while, some little tick is going to be on hyperdrive and its OS proteins are going to work really fast, and we may get it in the 24-hour mark rather than the 48 or 72-hour mark. But overall, if you keep your pet on one of those approved tick preventions, the tick will die before it gets a chance to transmit Lyme. Now, it does have a chance, other ticks, to transmit other diseases like Rocky Mountain spotted fever. But in general, those preventives are, especially if it's one of the newer classes like the isoxazolines, we think of Simparica Trio, NexGard, Bravecto, that group of chewables, they're usually very at working to kill the ticks. And so our chance of pathogens being transmitted goes way down. Okay, the small mammals who serve as host for the first tick life stage, can they transmit the disease? If a larval tick can pick up the disease from an infected small mammal and pass that on, if that's what you're asking. The mammal itself wouldn't transmit the disease, but the tick that feeds on that mammal, that for like the mice and all, absolutely could. Most of the animals we think of as reservoirs for Lyme in the wild are small mammals. They're usually rodents. Do we have any other questions?
Anything else from the audience, a question or comment, or if you have, if you do work in Lyme, anything else to share? Looks like there's one more comment. Equine protozoal myoencephalitis, EPM, as you know, it causes spastic limb. It is spread by exposure to feces of possums. Sarcocystis neurona, I think was what Jeffrey was thinking about. He was just
Yeah,
asking. Yeah, go ahead.
no, absolutely. Sarcocystis neurona, another one that's a fantastic one to talk about, less zoonotic concern. Um, and please don't kill your opossums just because they're in the area. If you have your horse feed properly stored and sealed, the opossums won't poop in it and it'll be okay. They do a lot of good in our environment. But yes, Sarcocystis neurona causes EPM, and that can be a progressive neurologic disease in horses. We typically treat it with the benzimidazoles. Now we treat it with a drug called Marquis or Panazaril. It's very effective. Um, but there was kind of a boom of people diagnosing it, and then it kind of trailed off. Um, but yeah, we do still see it, and it can cause that spastic paralysis.
I was wondering, is the parasite caused the damage, or is there something in between, like you have a bacteria, like you have in this disease, like Lyme?
Yeah, for EPM, it's both the parasite and the immune response that are causing the problem. So the parasite, when it's reproducing asexually, it can cause massive numbers of the parasite, and that is usually occurring in the central nervous system, which is a problem. And the immune response to that, the horse's body trying to fight it off, can also lead to those symptoms that we see. So it doesn't really have an in-between. There's no like secondary bacterial infection we're worried about, but EPM itself causes those neurological signs.
There's a human disease. This looks like the horse disease. No one knows what causes it.
That sounds scary.
Primary lateral sclerosis. And I've had a patient who has kind of worked in the garden a lot and got that, and couldn't find a way to see if they might be related or anything.
Yeah, did that
Something
patient
like that.
yeah, did you have any like lumbar sampling or CSF sampling in that patient to see if we had eosinophilia?
We didn't see that and didn't see the parasite in the CSF, and there's not any of the immune testing. There's just the test for horses, but couldn't find it possible for humans.
Okay.
I'd love to hear you talk about the thing you're mainly working into, if you want to quickly talk about. We're out of time. I see some toxoplasma brain tumors sometimes, but
Yeah.
they're about there.
My next question was going to ask if you had tested her for toxoplasma. So my work in toxoplasma is, I think, quite fun and cool. So we normally think of Toxoplasma gondii. It's a little protozoan parasite. Its main host is cats, both wild and domestic, and it likes to live in the GI tract of those cats, pass out in the poop, and then other people, animals, can get infected via ingestion of the oocysts that have passed out. They can also get infected via contaminated meat sources or the oocysts on vegetables, a lot of different ways to get infected with toxoplasma. We knew that pretty much any mammal can get infected. We knew that birds can likely get infected, but we don't know much about birds being infected with toxoplasma. And so my research was doing a prevalence study on waterfowl, primarily mallard ducks and quite a few other species across the US, at least hitting 3 of the major flyways that the ducks fly down. So it was a big effort where we had wildlife agents and hunters sending me duck hearts in the mail, which was quite an exciting thing to open up a box full of kind of stinky duck hearts in the lab. And then I would test them via PCR and via the modified agglutination test for both antigen and antibodies of and against toxoplasma. And so we found in the ducks that their prevalence ranged between about 20 to 40 something percent, which was pretty high. But we didn't know much past that. So the next step of my dissertation was to do an experimental infection where I actually raised up the little Toxoplasma tachyzoites, or the fast stage that we find in the blood, inject that intracelomically, so into the abdominal area of the ducks, and then measure serial antibody titers week by week, watch the ducks for any clinical signs, and then at the very end, humanely euthanize them and look at all of their tissues. So where we are with that is we didn't see any clinical signs in any of the ducks. We had different groups that we inoculated with high pathogenic strains in higher numbers and low pathogenic strains in higher numbers, and then the same in lower numbers in our control group. None of the ducks got sick. They were all very happy, healthy ducks. Antibody titers, we did see rather reflect the curve that we would normally see of became infected, antibodies went up, antibodies kind of leveled out, but never really went away. And we saw higher antibody responses in the higher virulence groups. And then histologically, we have only found burdyozoites in a few of the ducks in their brains. So normally we think of Toxoplasma as going to the brain, sometimes the lungs, and it likes to hang out in the muscle where it kind of goes quiet, waiting for a predator to eat it. But in these ducks, it does look like they go mostly to the brain. So currently, right now, as I'm finishing up work to get into my dissertation, we're going to be doing real-time PCR and looking at least 13 different tissue samples from each of the 60 some ducks that we had inoculated. So more to come on that.
All right. Well, thank you so much, everybody, for coming and listening to the talk. Thank you, Dr. Szafranski, for talking to us. And I know she's put her email right here. So if you all have follow-up questions or anything else, I'm sure she'd be happy to talk with you. But hope you all have a good rest of your day, and we'll see you next month.
Yeah, thank you. Check your crevices.
Absolutely. Thank you.
Yeah.